Hi, my name is Evan Seitz, and today I'll be showing you a short tutorial on how to use the SEAM GUI. And we're going to start at the SEAM repository, which can be found at my name, Evan Seitz, SEAMNN. And I'm going to assume that you've already seen my previous video on how SEAM works, and that you're now ready to dive in and actually get your hands dirty. And to help with all of the outputs that SEAM produces, I've created this SEAM interactive interpretability tool, which is a graphic user interface that allows you to navigate the data that SEAM outputs in order to really understand the different mechanisms and sequence determinants driving that variation. And so the first thing that you're going to want to do is download Anaconda and create this SEAM GUI Conda environment following these instructions and the important notes. And then you can come up to the SEAM folder after you've downloaded this repository. And inside of the SEAM GUI demo, you'll see that there's already a demo data set that I'm going to be using today. And this demo data set is based off of running SEAM on a fly enhancer from the DeepStar test set, this index right here, with 100,000 sequences along with their functional relationships formed from this one test set. So we're doing a local library with partial random mutagenesis and creating 100,000 attribution maps from DeepShap. And then there's an option in the GUI to either run with an embedding to cluster or to do something like a linkage matrix. And today I'll be focusing on the embedding. Okay, so I'm going to assume that you've downloaded the repository and now you're in the folder with the SEAM GUI here, and you can just Conda activate SEAM GUI once it's already been installed. And now we're just going to Python seamgui.py. This is going to open up the first tab. The GUI has 3 different tabs that you're going to be navigating through. And first we're going to import this MAVE dataset, which is our pandas CSV data frame, which is just sequences and their DNN activities, 100,000 of them. Then we're going to be putting in the 100,000 attribution maps via DeepShap. And here you can see that we're allowed some choices here. If we would have actually done our clustering or wanted to do our clustering on cropped versions of these maps for a higher resolution view of a certain mechanism, we can change that here as well. We can also change the reference sequence that we're using. And this is most important for a local library. And then in the embedding region, we can put in our UMAP file. And we're going to stick with k-means with 200 clusters. And we're going to be sorting our clusters by their median DNN activity. And we're going to select background separation because we are doing a local library and confirm our inputs. And now what this is going to do in the background is it's going to pre-process everything that we need to fluently use the GUI in real time. So we have one kind of a 1-minute bottleneck where we are literally processing 200 different sequence logos and different matrices that are required, doing the background separation, and also pre-computing all of those matrices as well. And this just allows us to not be interrupted when you actually want to go and try to dive into the analysis. Okay. And after this, we're going to be generating the summary matrix, and everything is complete, processing complete. So we can go back and we can see that it's automatically put us on this predefined clusters tab. I'm going to come back to this tab after I show you the custom clusters tab, which is only available if you put in a low-dimensional embedding like we have. And in this main figure, what you can see is that there are 100,000 points. Each one of the points in this space represents a single attribution map that has been embedded. And they are colored by their DNN score. So this is from DeepStar, just running the sequences through this. These are their predictions. And you can do a lot of things first to this representation. You can order by ascending, so you can see the highest DNN score points on top, or descending to see the lowest ones down here. You'll already see that there are a lot of trends going on across these islands. And you can also go and you can do things like arrange by Hamming distance to the reference sequence, or a histogram to show you where all of the points most often reside. And then you can go and you can click on here and form a small custom cluster. That's why the tab is called custom. And you can connect this and view the the cluster averaged attribution map and its sequence statistic properties in the form of a logo underneath. And so it's colored by orange with the reference, the wild type sequence that we can disable to see the classical logo here, but we can also change this from an enrichment to something like a PWM, etc. We can print out sequence indices to our terminal. This is showing us all of the different sequences that from our initial library that reside within this within this cluster. And we can do other things like we can I want to crop this view from 50 to 249, etc. Look at all the sequences that are actually in here in this table, and look up statistics that tell us about the what's going on inside this cluster. And all of this type of information is actually provided in the help section for this tab, and every tab actually has its own help section if you get confused at any point. So when we actually click on this page for predefined clusters, we can see that it has a very similar representation of what we already saw on this tab, except now the colors are the 200 different clusters that we defined on the imports tab for k-means 200 clusters. And what this allows us to do is to actually produce statistics over all of the clusters at once, which we can't do on the custom clusters. For instance, we can look at a box plot of DNN prediction over the sequences in each one of these clusters across this range, or we could view this in terms of the occupancy and so forth. And even further, the sequence dependencies for mechanism, which I'll show you in this cluster summary matrix. But first, I just want to get you familiar with how to use this tab. And what you can see here is that we have the logo. This is the cluster averaged attribution map for this cluster right here in black. This is the one that's selected. It's cluster 0, which can also be selected down here. Now I can toggle this to change the cluster automatically. You can see that I'm moving around here and seeing a lot of different things. Or I can go and actually click on the different clusters themselves and automatically see how things are changing as I move around. And so sticking with this cluster right here, or let's do, I guess, this one right here, cluster 9 of 199, we can also separate out the background. We can remove the background signal entirely to see what the foreground signal is, which is a TTK repressor, transcription factor binding site, and a GATA activator, and some curious stuff over here. And we can also go and we can view the average background on its own scale. So if I did fixed axis here, we'd see it's actually very small for this locus. Oftentimes it's comparable to the actual attribution signal. And we can also do things such as view the variability logo, which is an overlay of all the different cluster averaged attribution maps, just showing us where the poised transcription factor binding sites tend to occur. And when we're in this fixed y-axis, it's also very important to note just how small these features actually are, especially when we include background or not background, right? But that, as we start moving, we can see the relative change as we get to mechanisms that have larger and larger attribution signals. And this was the one that we looked at in the previous tab. So now I want to go and I want to use an example way down here. This is an interesting one because you can just see that the AP1 binding site changed from what it normally is. Almost anywhere else you look, the AP1 binding site is here, but down here it actually just moves over to the right. And so we can go and we can look at our cluster summary matrix here, and this is going to show us the different metrics. Right now we're looking at the positional Shannon entropy across our positions with respect to our clusters. And so let's look at that cluster 185. We can zoom in with this toolkit right here, the magnifying glass, zoom into this region right here. And we want to unclick our magnifying glass to actually let us now click cells. And what you can see is that at 185, which you can read in the top right-hand corner, it has 0 bits of entropy. And if I click this, you can see that for every sequence inside this cluster at this position, it's always C. That never changes, so it's 0 bits entropy. Likewise, if I click on this position right here, it's not that case. There's actually some bias towards being different nucleotides. And so you can also go and you can go to something like % mismatches to reference. And here you can see that this is actually very informative for why this de novo transcription factor binding site is occurring, and in our case, why is it shifting to the right. And you can see that there are two positions here that are creating this transition that occur at position 124 and 120. And what you can see is that even though the reference at this position is always C, we are never C. We are G or we are T at this position, so it's a 100% mismatch. And likewise over here, even though it's always T, we in this sequences in this cluster always have a C at this position, which is driving the mechanism. And so there are a lot of different things you can learn just by going through and viewing the sequence determinants. And this is really how the full sequence function mechanism relationship is decoupled using SEAM. And so that is an overview of how to use this software. If you have any problems running this, please leave a note in the GitHub issues, and I will see you in the next video.